Hello, welcome back everyone. I hope you all are having a great day. So today is Monday and hopefully everything sounds really good. Oh, that was the coffee pot going off. So I apologize. I was starting to go a little stir crazy in our home office. So sitting in the same room for 2.5 weeks for 10 to 12 hours every single day, including weekends, was honestly starting to make me feel a little stir crazy. And so I decided to move my computer and my recording microphone and everything else into our kitchen. So you might hear more noises in the background, like the coffee pot that just went off. You might be able to hear the bird more or the dog. So right now I have a lovely view into our great room, so I can see the dog sleeping on the couch. I also have a beautiful view outside and I can see our backyard and the green grass and all the dandelions that are starting to come up and all the weeds that I have to pull at some point. But other than that, I thought it was time for a location change just to get some inspiration and positivity back in my life. So again, I apologize if you hear more noises in the background because of that, but rest assured, they're just typical life noises. So I hope you all are excited. For this topic, we are going to talk all about transformation and oncogenesis. And this is one of my favorite topics. I think that it's just utterly amazing that this happens, and it's really interesting. So what we are going to do for this topic is we're going to start with transformation. So we're going to introduce the idea of what is transformation. We're going to talk a little bit about cancer and how does that happen. We're going to introduce the cell cycle, so hopefully that will be more of a reintroduction and that you recall many of the pieces of the cell cycle. We'll then talk a brief history of viruses and cancer, and then at the very end we're going to take everything and we're going to put it all together, and we're going to answer these questions of how do viruses change cell signaling transduction pathways, how do they disrupt cell cycle control pathways, and all of this is in an effort to better understand how viruses can transform cells and how they can cause cancer. So we're going to start with just a couple of tables. So both of these tables on this page show us the viruses on the left, and then on the right we have their associated cancers. And this is not an exhaustive list by any means, but this just gives you a flavor that there are both RNA viruses and DNA viruses that are associated with cancers. So when it comes to viruses and their involvement with cancer, in general, about 20% of cancers are somehow related to viruses. And of course, that is going to be 20% of all human cancers. So let's go ahead and first kind of talk about some key points here. So when we think about viruses and their involvement with cancer, it's very important to, number one, say that viruses themselves do not cause cancer. So oftentimes when people talk about viruses and cancer or oncogenesis, oncology, if you will, they say, well, viruses cause cancer. That's not necessarily true. So viruses themselves do not cause cancer. In turn, they cause changes in cells that lead to cancer. And really, I should say that can lead to cancer, right? Not all changes will necessarily lead to cancer. And really, we can say that viruses are unwilling initiators of oncogenesis. And the reason that we say that they are unwilling initiators of oncogenesis is because this idea of transformation and oncogenesis is not a requirement for viral propagation. And I'm going to put an asterisk there. And the reason for that, of course, is there is an exception to every single rule. So although transformation and oncogenesis is not something that a typical viral reproductive life cycle is needed, there is an exception to this. And the exception to this is a walleye dermal sarcoma. And the walleye dermal sarcoma is a really interesting story. And so with this virus, there is both tumor production and regression. And this tumor production and regression is essential for its reproductive cycle. So right now, this is the only virus that we know of that is an exception to this rule, if you will. Suspect there will be other ones in the next coming years as we learn more and more about viruses. But one of the really cool things with this walleye dermal sarcoma is that it appears to be seasonal. So the fish will get infected during the spring, and then through the fall and the winter, the tumors will grow, and then actually during the spring, it appears that regression happens because viral expression changes. And so it has to do with when the fish would be more active and whatnot. So it's kind of interesting that this virus has evolved to both do tumor production and regression, and that it is essential for its reproductive cycle. So oncogenesis is accidental, and that is a key point to remember as we move forward in this lecture. And of course, when I say oncogenesis is accidental, there is that asterisk there again with the walleye dermal sarcoma. So with that virus, it is definitely absolutely necessary for its reproductive cycle. But other than that exception that we know about, oncogenesis is accidental. So this idea of how viruses can cause cancer, or how viruses even became associated with cancer, really began with studies of cultured cells. And so that leads us to this idea of transformation. So when virologists were working with cell lines, they noticed a couple of things. And the main thing that they noticed was that when some viruses infect cells, there can be both morphological and growth changes. So this idea of viruses causing morphological and growth changes, changing these growth properties, we call this idea transformation. So they called these cells transformed. And again, all of this began in a petri dish as we were growing cells. So let's talk a little bit more about, well, what are some of the properties of these transformed cells? So as virologists were looking in these petri dishes of cells, what else did they notice? So one thing that we have is we have immortality. So these cells were immortal. They grew indefinitely. The other thing that they noticed was that there was a reduced requirement for growth factors. So typically, a cell is going to sense its environment, and when growth factor is present, that is a signal that the environment is conducive to growth, and the cell will start growing, replicating its DNA, and going through the cell cycle. So one of the things that we see in transformed cells is that they kind of ignore some of those signals. And so that leads us to this idea of messing up of signaling pathways. So when we have messing up of signaling pathways, we can get growth without the signal to grow. We can also have problems where these cells grow to saturated amounts. And so if in a typical dish, when we're growing cells, they tend to grow to a confluent lawn and they evenly grow out. They don't tend to grow on top of one another. When we have cells that have been transformed, one of the things that we start to see is that they start to grow on top of one another, and they cannot sense their environment. And so that's part of this idea of signaling pathways being broken as well. So again, they're going to be growing over one another, so growing on top of their neighbors. In terms of morphology, morphologically, they look different. So if they are normally round cells, Maybe now they're more elongated. So they definitely have issues with the typical normal things that we would expect from a cell. They have a loss of capacity for growth arrest upon nutrient deprivation. So even when a typical cell, if we deprived it of nutrients, would slow growth, this doesn't happen to transformed cells. So if we think about a cell that has receptors on it, and it's going to have different signal transduction pathways that are going to occur, those will not normally happen. So normally when you would get a signal to stop growth, they're going to ignore that and instead they're going to grow. So again, these are things to be thinking of, and we'll introduce some of the signaling pathways as we move along and which ones are altered. So when we think about all these things, all of this actually goes back to the cell cycle. So before we jump into the cell cycle, let's introduce the idea of cancer and kind of talk about what is cancer. So if you ask most people what cancer is, most of them say that it is uncontrolled growth of cells. But my question to you is, well, is uncontrolled growth of cells enough to cause cancer? And the answer to that is a terse no. So if you are more interested in some of the details that we aren't talking about in this today, we actually do an entire topic over a couple of days in cell biology. So if you've taken cell biology, you'll have a little bit more insight. And I know many of you I will see in the fall. So we will come back to some of these ideas. So it turns out that this idea of uncontrolled growth of cells is not enough to cause cancer. And that brings us to this idea of the hallmarks of cancer. So this was and still is one of the most heavily cited papers in Cell. And what it does is it starts to tie in some of the other things that are the hallmarks of cancer. And so one of the things that you'll see, let's go ahead and let's start here on the bottom. So we see enabling replicative immortality. Well, that sounds familiar. Doesn't that sound like the idea of transformed cells being immortal and growing indefinitely? So we will see enabling replicative mortality when we have cancer. Inducing angiogenesis. Angiogenesis deals with blood vessels. And so we'll get more blood vessels that will grow throughout the tumors and to tumors to help supply blood and nutrients to it. So resisting cell death. So of course this is going to be stopping that idea of apoptosis. On the top here we have sustaining proliferative signaling. And so this is having a keep growing signal. And we'll talk about how these things are broken. Evading growth suppressors. So we typically will have growth suppressors, signals that tell the cell, hey, you should stop growing. And so this is essentially like ignoring the stops. So if you have a stop sign and you decide to not stop and just roll right through it, that would be evading a growth suppressor. We also have activating invasion and metastasis. And this deals with growing in other places. And this is a very key hallmark of cancer that we have. And so typically in a cell, we would have a signal that says grow, but we would also have maybe another signal that says stop growth. And so we have these combination of different signals in the cells. Well, if you have a cell that has cancer, it doesn't need to listen to this grow signal. That cancerous cell, so let's go ahead and make our cancerous cell here in pink. And actually, I'm going to make it a different shape. It may or may not have a receptor, but there may not be a signal that says grow, and that cell will grow. In the case of the stop growth, it might have a stop growth signal, but instead of stopping growth, it will continue to grow instead. So those are examples of not only sustaining proliferative signaling, but also evading growth suppressors. So over the years, we have actually added more hallmarks of cancer, and some of the other hallmarks of cancer that we have added, so I'm going to try to squeeze some of these in here on the bottom of the page, are abnormal metabolic pathways. So we actually see that in many different cancers, we have different metabolisms that are used by those cells. There's an entire field of study that is actually looking at what are the metabolic differences in different cancers, and can we somehow use those metabolic differences as treatment. We also have things like avoiding the immune system. So a first part of our immune system's job is to help clear potentially cancerous cells. We also have things like genome instability. We'll oftentimes see multiple copies of chromosomes, so more than just the 2 copies or parts missing. We'll also see things like inflammation. And again, there are many other hallmarks that have been added, but I did want to give you a little bit of other ones that are not listed in this Hallmarks of Cancer that was originally developed by Weinberg and Hanahan. So now that we've talked a little bit about what is cancer, what are some of the hallmarks of cancer, let's talk more about how do cancerous cells differ from normal cells. And we've actually kind of started hinting at this. So one of them is this idea that the genetics are messed up. And so again, you might have many copies of a chromosome. We can talk about size and shape, and just as a whole, morphology tends to be different. We said metabolism. We tend to, in cancer, actually see more anaerobic pathways. We see a different proteome. So pretty much any of the omics that you can think of, metabolomics, proteomics, genomics, all of those are going to be different between a normal and a cancerous cell. And actually using all of the different omics is ways that we've learned more about how cancerous cells differ from normal cells. And we already mentioned this idea of altered signaling pathways, and then the cells that they were originally, they're going to differ from those. They are going to continue growing, and of course we talked about growing on top of one another. So that was a hallmark or a property of transformed cells. This is also something that is a property of cancerous cells as well. And so again, this idea of having these weird clusters that form, and it's because they cannot sense their environment. So going back to those altered signaling pathways. So thinking about cancer, we have to talk about cancer critical genes. And oftentimes these are called oncogenes. And so we're going to walk through what these 3 types of cancer critical genes are, and then we're going to talk about the cell cycle. And we're going to try to interweave these pieces together. So the first type of oncogene that we're going to talk about is a proto-oncogene. So for a proto-oncogene, this is going to be a gene that normally promotes cell growth. And so this is going to be a gene that says, hey, turn on growth. And when we think about cancer, we're going to have a mutation where the proto-oncogene is on. And if there's a mutation where it is always on, that could lead to one of those hallmarks, which is sustaining proliferative signaling. And so as we talk about each of these genes, we'll actually talk about whether they would be on or off in the case of cancer, and I'm going to try to color code things so that you all can keep track of it. So the next type of gene that we have is a tumor repressor gene. So for tumor repressor genes, these are going to normally restrain growth. And so they're going to stop growth in the case of maybe nutrient depletion, or perhaps during copying of DNA, we made a mistake and it's irreparable, and so that cell now has to undergo apoptosis eventually. So stopping the cell from continuing going through the cell cycle would be considered a tumor repressor gene. So in cancer, we would see a mutation where repressor genes are off. And the third type of cancer critical gene category that we have are genome maintenance. So for genome maintenance genes, these are going to make sure that the integrity of the genome remains intact. So for genome maintenance genes, this is going to be when we have mutations where the genome maintenance genes are off. And again, this is going to be in cells, in cancerous cells. We will see genome maintenance genes that normally help make sure that the genome remains intact. So some of those genes that encode for proteins that are responsible for fixing DNA when there's a mistake, if we have a mutation in those genome maintenance genes that are off, that would be an oncogene. So these are the three types of cancer critical genes. And again, many different genes kind of fall into these categories. So let's go ahead and now switch our gears into thinking about the cell cycle. So hopefully you recall from Bio 185 and from genetics. And if you take cell biology, if you've taken that recently, we also talk about the cell cycle in that class as well. We talk about it a little bit more detail than this, but we're going to go ahead and just to make sure that we're all on the same page, because to understand how transformation and oncogenesis and how viruses relate to everything, we have to look at the cell cycle. So for the cell cycle, we have a couple of different phases. So I'm going to try to do my best to draw it here in a circle. So our first phase that we have is G1. So G1 is our first growth phase, and we're asking here, is a new cell needed? And if it is, we're going to go ahead and grow. So here we have, within G1, we have that checkpoint. So in that checkpoint, this is where the cell is asking, is a new cell needed? So that is part of that checkpoint. And if the answer is yes, then we go through the S phase, which is the synthesis phase. And this is where all of our DNA is copied. So we replicate all of the chromosomes. And then we enter another phase, another growth phase. And these are not drawn to scale, by the way. And that second growth phase follows the replication of our genome, and we have another checkpoint. And in that checkpoint, we want to make sure, is all of our DNA replicated? So have we fixed any mistakes? And this is where those tumor repressor genes, those genome maintenance genes, are going to come into play. So is all of our DNA replicated? We're going to make sure that all the chromosomes we started with, all of those are replicated. If we made any mistakes in the genome maintenance genes, we're going to use their gene products, those proteins, to help fix any mistakes at this point. And if we pass that checkpoint, we of course have the very last part, and that M stands for mitosis. And so in mitosis, the cell is going to go ahead and divide. Turns out in mitosis, we also have a checkpoint. And the checkpoint that we have is the spindle checkpoint. And if you recall, one of the last things that we do in mitosis is we actually pull the chromosomes apart so that each of the daughter cells gets each of the copied chromosomes. So we want to make sure that each cell is identical, and so there is a checkpoint to make sure that the spindles that grow from the centrioles, that each of them are attached to a sister chromatid so that we don't accidentally end up with one cell that has more chromosomes and one cell that has less. So this is our cell cycle, and what I'm going to do is I'm going to zoom in here just so that I have a white space here to work with. And if you're wondering what this has to do with our oncogenes that we just talked about, I kind of want to quickly illustrate that. So let's go ahead and just draw a mini cell cycle here. So G1, S, G2, move this more in the middle. All right, so here we have just another little cell cycle, and on the top, kind of feeding into the cell cycle, we have a go signal, and that could be our proto-oncogenes. So if we have a mutation that turns them on indefinitely, you're always going to be going through the cell cycle. Now, on the flip side, we have our tumor suppressor genes. And again, if you have a mutation, it's going to stop these. So normally tumor suppressor genes, if there's a problem, are stopping the cell cycle. But if we have a mutation in them, then they're no longer going to be able to do that. So again, helping you kind of put the pieces together of how the cancer critical genes and the cell cycle kind of feed into one another. All right, so let's go ahead and we need to talk about two important signaling pathways. So we've introduced the cell cycle, we've introduced what cancer is and what the cancer critical genes are, but in order to fully understand everything, there's two signaling pathways that we're going to talk about. One of them is RB, and RB is retinoblastoma protein. The second pathway is P53, which is also a protein. So we're going to walk through both of these signaling pathways, and both of these involve proteins. So let's start with RB, or the retinoblastoma protein. So for RB, we're going to draw what no cell growth looks like versus cell growth. So let's do no cell growth on the left, and then we're going to talk about how that goes to cell growth. And I'm going to write RB up here so that we know that this is the RB pathway. So normally in our no cell growth, we are going to have the RB protein, and RB is going to hold transcription factors. So if you have transcription factors that are held by RB, and then in the nucleus, so first we have our DNA, we're going to get no transcription. And so this is going to be off. So a lot of the things that we need to move through the cell cycle, those are going to be off. So now, as we transition from no cell growth to cell growth, we're going to ask the question of ready to divide, right? Is the cell ready to grow? Is a new cell needed? And if the answer is yes, what will happen is we will have our retinoblastoma, and this retinoblastoma protein will be phosphorylated. And when that retinoblastoma protein is phosphorylated, it releases the transcription factor. And when these transcription factors are released, I don't know why, but I really struggle with drawing double-stranded DNA. So when those transcription factors are released, they can of course go and interact, and we'll have RNA polymerase and all the other good stuff that we need for transcription to be turned on. So now we have transcription that is turned on, and this is going to help advance the cell cycle. So a lot of the pieces that we will need for going through the cell cycle will be produced. So for RB, we can classify this as a tumor suppressor protein. And so this is normally going to restrain growth. If it is damaged or if its regulation is affected, it cannot do its job. All right, so let's go ahead and now talk about the second important signaling pathway, and that is P53. So P53 is a tumor suppressor gene, and we're going to talk about what happens if we have DNA damage. So let's go ahead and draw some damaged DNA up here. So here is DNA damaged. Now, normally when we have DNA that is damaged, we have P53, and P53 can do one of two things. It can stimulate DNA repair, and so the cell will try to repair the DNA, and when it does that, it will stop cell division, which makes sense, right? We don't want to continue on with the cell cycle if we have damage in our DNA. So it will stimulate or turn on DNA repair. If DNA cannot be repaired, P53 is going to turn on apoptosis. So it kind of has multiple roles here. It'll turn on DNA repair, turn off cell division. If DNA cannot be repaired, it will turn off cell division, turn on apoptosis. And so you can imagine that if P53 is broken, then DNA repair will not happen. So not only will DNA repair not happen, but apoptosis won't happen, and the cell will continue to divide. And if that happens, of course, then whatever was damaged with that DNA, that is going to be passed on there. So now that we have all of the pieces of these different signaling pathways and the cell cycle, we're going to pause this for a second, and we're going to turn our attention to history. And if it seems like we're kind of talking about things in different chunks, we are for now, but hang on with me. When we get to day 2, we will actually start putting all of these pieces together, and it'll make more sense why we've kind of chunked out these pieces here at the beginning. So let's talk a little bit about the history of viruses and cancer. So in 1908, so Wilhelm Ellerman and Olaf Bang. So what these two scientists did was they looked at sick birds that had leukemia, and what they then did was they took filtered extracts from the bird. So what I mean by that is that they took extracts and then they filtered them first. And so they knew that they had viruses. So they knew that these birds were infected by viruses, and they saw that these birds were getting leukemia. And so they thought that they were somehow related to one another. So they then took extracts from the birds. They filtered those extracts because, recall, at this time, we still had the definition or the working definition that viruses were smaller than 0.2 microns, because we would use 0.2 microns to filter sterilize things, and anything smaller was considered a virus. So then what they did was they put that extract into a healthy bird. And you can probably guess what happened to that healthy bird. That healthy bird then got leukemia. So the problem was that at this time, leukemia was not recognized as cancer. So leukemia is a cancer of the different blood cells. And so at the time, cancer was thought that you had to have a tumor. And of course, with leukemia, you do not get a tumor. And so at the time, the scientist's work was not recognized as cancer, and people kind of just honestly ignored it. So around the same time, we had Peyton Rous. And what Peyton Rous did was he actually studied chickens. So in 1909, he began his work, and farmers brought him chickens with tumors on them. And so the image here on the left, this is Peyton Rous. So on the right here's a chicken, and here is a tumor that the chicken had. And so his big question that he had was, well, what caused the tumors? So that was his question. And what we're going to do is we're going to start with a blank page here, and we're going to talk about his experiment that he did. So his question was, where did the tumors come from? What is causing those tumors? And so what he did was he took a tumor, and he took this tumor from an English hen. So he took it from a chicken, and he ground it up. So he made an extract, and he filtered it. And again, the thought was here that viruses maybe have something to do with this. And then what he did was he injected the filtered extract into a healthy chicken, which you can probably guess what happened to that healthy chicken. That healthy chicken then of hers got cancer. And so we say, I'm going to put first here in quotations, we say that he was the first to show that cancer can be caused by a virus. So Peyton then named the virus Rous sarcoma virus after himself, or RSV. And RSV, as we later went on to learn, is a retrovirus, and it's the first oncovirus to be discovered. So he showed that that cancer can be caused by a viral infection. So at this time, people didn't really believe him. They didn't accept that this was a thing at all. And if we kind of fast forward a little bit into the future, so in the 1950s, when we think about cancer researchers and what people thought at the time, there were definitely split camps there. So we had first the virologists, so like Peyton Rous there, that said that viruses cause cancer. Of course, the problem was that this was not shown in humans yet. So we showed this in birds, but we didn't show this in humans yet. So the virologists were on the side of viruses cause cancer. We of course had our epidemiologists, and epidemiologists said that exogenous chemicals cause cancer. And of course the problem with their hypothesis was that there was no mechanism. So even though they thought that different chemicals could cause cancer, they hadn't really told the scientific community how they think that. And then we have other, which we can think of, are molecular biologists or geneticists, and they thought that genes cause cancer. And specifically, they were thinking about mutations. And the problem was they had very weak evidence. Now, of course, moving on to 2020, you and I now, of course, know that they're all right. So even though people kind of set up camp and there was a huge scientific debate over what happened, we, of course, know that all three of these groups were correct. Exogenous chemicals can cause cancer, mutations in those genes can cause cancer, and, of course, viruses can also, though accidentally, cause cancer. And so kind of moving along in history, we have another scientist, Howard Temin. So hopefully you have recognized both RSV and the name Howard Temin. So when we first started talking about how we do reverse transcription and integration, we actually introduced both Temin and Baltimore. So both Temin and Baltimore won the Nobel Prize for, of course, their discovery of reverse transcriptase. So he was one of the scientists that won a Nobel Prize for that. And I did not mention it when we were talking about Peyton Rous, but he did actually eventually receive the Nobel Prize for his work. It was much, much later, but long incubation time there for that Nobel Prize, but he did win a Nobel Prize for his work. So let's talk about Howard Temin. He worked in the lab of Delbecco. So we've talked a little bit about Delbecco. So he developed, I should say, the plaque assay for eukaryotic viruses, and he, of course, his name is on a lot of other things, but Temin worked in his lab, and he originally actually started working with fruit flies. He got a little bored, and he joined, of course, the good side, the side of viruses. And so what his goal was, he wanted what he referred to as cancer in a dish. So at the time, everyone was working with chickens or other birds, right? They were making these extracts, they were infecting the birds were getting cancer because of the viruses, and things weren't really moving forward. And so what he wanted to do was he wanted to be able to work with the cell line that had cancer so he could have cancer in a dish. So because Rous sarcoma was only shown to cause cancer in live chickens, he wanted to recreate it essentially in the lab. So what his experiment was he took normal cells, and you can take cells from different things. You could do a biopsy. At the time, people would take other organisms and kind of remove the really bony parts, mash up the rest of the cells, and then make extracts from those, and then start cell lines that way. So he took normal cells, and he started his cell line, and what he did was he added RSV, so Rous sarcoma virus, and eventually what he got was he saw that the virus transformed the cells. And so he saw tumors, he saw immortality, so all of the other characteristics that we see with transformed cells. So he was able to succeed there and went on to do a lot of other experiments with these cells, and of course this helped to kind of change the road of virology as well. So in terms of RNA retroviruses, which is what RSV is, it kind of makes sense, right? So if we think back to the life cycle of these, we talk about how they reverse transcribe and they integrate into DNA. So, all right, this sort of makes sense that these RNA viruses have been implicated in cancer. But as we kind of move forward in history, we learned that DNA viruses can cause tumors too. And so if we take a look at the two images that we have here, we've got these rabbits, and both of them have, you can hopefully tell that they've got these protrusions, and these protrusions are caused by papillomaviruses. And so in 1933, papillomavirus was discovered, and it was the first DNA oncovirus, and this was shown in cottontail rabbits. Okay, so like I said, in 1933, the first DNA oncovirus that was discovered was papillomavirus, and just like Temin succeeded in essentially making cancer in a dish by infecting cells with Rous sarcoma virus, we did something very similar with DNA viruses. And so what scientists were able to then do was, with cell lines, scientists were able to show that papillomaviruses and polyomaviruses, and so thinking back to SV40, and so they showed that they can cause cancer. And so we are beginning to get more and more evidence that viruses are associated with cancers. So this kind of wraps up our history that we're going to talk about for viruses and cancer, and this will be the end of day 1. When we come back for day 2, what we're going to do is we're going to take all of the pieces and we're going to put it all together. So right now, I've kind of given you brief things that scientists develop or have found in history, how we discovered oncogenic viruses. I haven't fully told you all of the details of how they're actually identified, but we're going to talk more about that when we come back in day 2. So we're going to put all of the pieces together from parts 1 through part 4. So thanks for hanging out with me, and I will see you on the flip side. Bye, everyone.